Hi guys, it's me, your biology teacher. Today we are going to study about the plasma membrane of eukaryotic cells and with some exceptional changes within the prokaryotic cells. Okay, so we're going to start with the definition. The first thing, that how we can state that what is the plasma membrane itself. So we can say that it is the outermost boundary of an animal cell while it lies next to the cell wall in plant cells. So this is the easiest definition that we can define the plasma membrane. All right, students? So now, if we move forward, we should know about who discovered the plasma membrane and what kind of structure it exhibits, or what we can say that the plasma membrane looks like under the compound microscope and the electron microscope. Okay? So talking about its structure, different scientists, they worked on explaining the structure of the plasma membrane, but the thing is that in the ancient times there were no microscopes. But later on, with the invention of the compound microscope and later on with the invention of electron microscope, scientists were able to explain about the structure, that what the plasma membrane is made up of, what kind of molecules even they are present inside the plasma membrane. So the first thing is unit membrane model. Okay, so the unit membrane model explains that the plasma membrane just looks like a sandwich. That's why sometimes some scientists say that the unit membrane model can also be called as the sandwich membrane modeling. Okay, now what does it explain? If you look at this image, guys, okay, this image explains that the plasma membrane is made up of two backbone layers made up of phospholipids and that are covered up by the protein layers on the upper side and on the lower side. Okay, so just look at this structure. That's why it is known as the sandwich membrane model, because these cellular balls that you can see in here, okay, with the tails, they are known as the phospholipid bilayer because they are made up of two layers. One layer lies upward and the other one is present on the downside. And later on, it was observed that the proteins, they cover up these phospholipid bilayer molecules. So that's why this is known as the sandwich membrane model, okay? Now, if we talk about it a little bit in more detail, it was explained by different scientists that all the organelles within the cells exhibit the same structure. Okay, now the question is, what does this statement means? That all the structures they exhibit, all the organelles they exhibit the same structure. What does that mean? It means that all of the membrane bounded organelles present inside the eukaryotic cells, they have the same phospholipid bilayer backbone inside all of their internal or external membranes. All right, students, so this is the sandwich membrane model or the unit membrane model, which was explained first of all, and it was a kind of, you can say, comprehensive explanation about the unit membrane. I will explain later on presented more accepted model that is known as the fluid Mosaic model. Yeah. Now, the fluid Mosaic model, if you just focus on the statement, there are two different words that could be new for you about the plasma membrane. The first one is explaining fluid. Okay, it means all of the plasma membranes, including the internal membranes of the organelles, they are dynamic. Okay, they are showing us a kind of simple movement within their own premises. It means what? They stay on their place and they're all the time moving. All right, so they are dynamic membranes. Now the second thing is mosaic. Mosaic is what? It's a mixture of different types of molecules. A mixture of different types of molecules means what? The phospholipid molecules themselves, the fatty acid tails, and number three, the proteins that could be present embedded inside the phospholipid bilayer, and those proteins that could be floating on the surface of the phospholipid bilayer. So these different types of molecules that are different in their chemical composition, that are different in their textures, and that are different in their organization, they make up this mosaic of the plasma membrane. Okay, this model, it was proposed by Singer and Nicholson, two well-known scientists. If you have studied about the movements of the particles across the plasma membranes and so on, you should be familiar with them. Okay, now how we can state this kind of model? We can say the proteins are not continuous on the surface of phospholipid bilayer. Instead, they are embedded inside or float on the surface like icebergs. All right, students, so this is the statement about the fluid mosaic model. It can explain the whole structural composition of the plasma membrane, that how does it look like under the electron microscope. Okay, so this is your fluid mosaic model. Now, if we move forward, we can say that what are the different functions of plasma membrane? All right, so what kind of functions could be performed by the plasma membrane, that is the outermost boundary? Number one, protection. All right, just like the boundaries or the gates of a building, they are supposed to protect the internal components. Just like that, the plasma membrane is the outermost boundary. So it should protect the internal components like the cytoplasm, like the mitochondria, Golgi bodies, nucleus, and other organelles as well. Okay? The second function is about the transport. Now, for our understanding, I'm going to divide the transport into two basic types. The first one is passive transport. Okay? And the second one is active transport. All right, guys? Now, who can differentiate between these two different types of transports? Number one is the passive. Yeah? Go ahead. Number one. Passive transport is the movement of particles from an area of higher concentration to an area of lower concentration along the concentration gradient, and no energy is needed in this case. Okay? This is your passive transport. Now, the familiar type of examples that you have studied almost hundreds of times in your previous classes, diffusion is a type of passive transport. Osmosis is a type of passive transport, okay? But for the osmosis, I'm going to change the definition a little bit, like how we can define osmosis. Specifically, we can say the movement of water molecules from an area of higher concentration to an area of lower concentration across a selectively permeable membrane. All right? So this is osmosis. Then there's a third type that is known as facilitated diffusion. Okay? Now, what is facilitated diffusion? To facilitate some particles that cannot cross the plasma membrane, but here are some proteins that are known as permeases. They're going to aid them. They are going to help these particles to move from the higher concentration to lower concentration. Because this is, again, even if it is facilitated, this is a type of diffusion. So the particles going to move from higher to lower concentration. Okay? Then, the active transport, it's totally different from the passive transport. Okay? We can say that the active transport is energy demanding type of transport. Like the particles, they can move from their lower concentration this time, and they're going to move to their higher concentration against the concentration gradient, and this is active transport. Now, the question is, in what form normally the cells, even if they are the plant cells, they are the animal cells, or even if they are the microorganism cells, in which form they use their energy? Oh, when the energy is needed by the plasma membranes to move the particles, in what form the energy is available? Yeah, that is a specific organic molecule, ATP. Okay, that is adenosine triphosphate. That adenosine triphosphate is primarily a nucleotide. Okay, but it is the energy currency of all of the living cells. Whenever the cell needs energy, it uses ATP. It breaks up one phosphate, uses up the energy of that bond, and the ADP is the leftover, that is adenosine diphosphate. That adenosine diphosphate is again sent back to the mitochondria, where it is again replenished, and it comes back at the point wherever the energy is needed. All right? So, these are the two basic types, the passive transport and the active transport. Now, students, if we talk about the bulk movements of the particles across the plasma membrane, we're going to move to a new type of movements. That is endocytosis. Okay? Now, what is endocytosis? Endo, just look at the term. If you understand the meanings of the term properly, you can define it by yourself. Okay? Endo means inside. And cytosis is the movement of particles within the cells. So when the particles, they move from outside environment of the cell towards the cytoplasm of the cell, this kind of movement is known as endocytosis. Yeah, well, we were talking about the endocytosis, all right, the movement of particles from outside the cell towards the cytoplasm, okay, moving the particles into the cell, that is known as endocytosis. Now the thing is, like how the cell bring the particles into the cytoplasm, okay, students? Now the important thing is the plasma membrane, it projects outward, okay, it engulfs the particles into a sac-like structure, and then that invagination, okay, that thing which brings the food particles in, that's known as invagination. Now that invaginated part of the plasma membrane, it just slightly pinches off from the plasma membrane and it drops down into the cytoplasm. Now their fate, it is depending upon the type of the food particles that have been engulfed, like whether if they are proteins, they are carbohydrates, or they are the fats or oil droplets. So they will be digested by their own respective specific enzymes, and these enzymes, they will split up these bigger particles into the smaller pieces that can be absorbed and used to harvest energy. All right, students, so this was the endocytosis. Now this endocytosis is again differentiated into two types. The first one is phagocytosis, and the second one is pinocytosis. Okay, these are two different names of the same type of processes. The first one is phagocytosis. The phagocytosis is the movement of solid food particles into the cell by engulfing them. Okay, this is known as phagocytosis. The second thing is pinocytosis. Pinocytosis is the second type of endocytosis in which the cell brings in all types of liquids. Whether liquids, they belong to the inorganic type of solutions, or organic types of solutions, or simple water. Okay, now the thing is, what about the bigger particles? Because in the beginning we said that the phospholipids, they make up the backbone of the plasma membrane. So if they make up the backbone of the plasma membrane, it means what? The organic molecules, they can simply diffuse into the plasma membrane without using any energy. Okay, so that was a kind of tricky thing about the fat molecules. Now students, we're going to talk about exocytosis. All right, now what is exocytosis? Exo, going out. Cytosis, movement of the particles. So when the different types of particles that have been digested and they can no more be used by the cell. Now what's going to happen to them? These particles, they will be packed up within the vesicles. Those vesicles will travel to the plasma membrane, and the plasma membrane will burst up. It's going to open up so that the food particles or the undigested waste products, they can be pushed out of the cell. This process is known as exocytosis. Okay, so this is a general review about the structure of the plasma membrane and the functions of the plasma membrane that are performed generally within an animal cell. All right, students, so this is the basic thing. Now I'm going to show you how the particles, they move in and move out, how the energy is used. Okay, so number one, just look at this video. If you just focus on it, you will understand like different points that I have just explained. Number one, those particles that need no energy to pass across the plasma membrane, they are straight away going through specialized proteins, and these proteins are permeases. Okay, they can be simple water molecules, they can be lipid soluble molecules, okay, or they can be sometimes ions that are moving from the higher concentration to the lower concentration. Now secondly, if you just look at it, some bigger particles that cannot pass across the plasma membrane, like through the pores that it exhibits, in that case these proteins, they're going to help it, and they will move them from their outer side to the inner side by providing special passages, by providing special channels so that it can move into the cytoplasm. Okay. Now the next thing is about endocytosis and exocytosis. Okay. Before the endocytosis and exocytosis, if you look at this special protein that seems bigger than the other proteins, this is what? Yeah, it's ATPase. This ATPase is what? It's a special kind of protein which is always using protons, and it is pushing the particles from its lower concentration to its higher concentration. like whether the particles, they are supposed to be pushed from inside to outside, or they are supposed to be pushed from outside to inside, they will be always pushed or pumped by these special proteins, and these proteins are ATPases, okay? Or they can be simply known as ATP pumps. Pumping means what? Moving against the concentration gradient. That's why we use this term, the pump. Now, the next part explains endocytosis and exocytosis that I have just touched in here. The particles moving in, endocytosis, and the waste products or undigested food going out, that is exocytosis. All right, students, so that's all for today's session.